And now to our second lecture on amines. Amines can also do conjugate addition, which we introduced back at the end of chapter 18. Thus, if I have some kind of carbonyl compound that has an alkene adjacent to it, this type of compound is called an alpha-beta unsaturated aldehyde or ketone. And I introduce an amine as a nucleophile. The amine can come in at the beta carbon, thrust these electrons up, and then protonate this alpha carbon to give you this beta-added aminoaldehyde. I now want to begin exposing you, and I mean that in a non-creepy way, to heterocyclic compounds, beginning with this slide. And what is a heterocycle? By definition, a heterocycle is a cyclic compound in which there is more than one kind of atom found inside the ring. For example, these three cyclic compounds are all heterocycles because each of them contains 4 carbon atoms and an additional atom other than carbon, either nitrogen, oxygen, or sulfur in the ring. There are lots of different nitrogen-containing heterocycles. One of these is pyridine, which you might remember from back in chapter 10. Because it meets all of the rules of aromaticity that we discussed in chapter 15, pyridine is aromatic. The nitrogen lone pair, which I haven't drawn here, is not released into the aromatic system because it is perpendicular to these pi electrons in the ring. These resonance structures here show us how the nitrogen in pyridine has a partial negative charge, which means that it actually withdraws or sucks electron density from the remaining carbon atoms in the ring. Now, because pyridine's nitrogen atom is electron withdrawing, electrophilic aromatic substitution reactions on pyridine occur at the meta position relative to the nitrogen. Thus, we could say that pyridine reacts similarly to nitrobenzene during an electrophilic aromatic substitution reaction. So here's our nitrogen in pyridine. I do an electrophilic substitution reaction. The electrophile will get added at the position meta relative to the nitrogen, similar to the way we would see in the electrophilic aromatic substitution of nitrobenzene. There are various other nitrogen heterocycles that I want you to have met. Not that I'm going to ask you any in-depth questions about them on an exam, but because I don't want them to look completely foreign to you when you come across them later in your careers. This compound, found at the core of numerous natural molecules, is called quinoline. Its structural sibling, shown here, is called isoquinoline. Now, isoquinoline should look familiar to those of you who are in my lab class because it is found at the core of this potent anti-leukemia molecule, AM636, which we discussed in lab as a potential group synthesis project. Note the isoquinoline core buried right here inside the molecule's lower left-hand ring structure. Another highly significant nitrogen heterocycle shown here is the porphyrin ring system. Why is it so important? Because this ring system constitutes the active site iron core of heme proteins, including hemoglobin, which our bodies use to transport oxygen to all of our tissues. Thus, we can see in the core of hemoglobin proteins, it has this heme active site in which these nitrogens present in the porphyrin ring system chelate an iron 2, which can be reduced or oxidized as it captures and releases oxygen from that we breathe. Because I don't want you to leave this class without hearing the word alkaloid, I am now going to say it with a funny accent. Alkaloid! Alkaloids are a special class of naturally occurring amines. A few famous examples of these are shown here, which you're welcome to remember if you so desire. This is an example, atropine, which is an antispasmodic alkaloid. This is a compound called ephedrine. And this is an antihypertensive drug known as reserpine. This brings us to one of the most important family groups of nitrogen heterocycles on the planet, the purines and pyrimidines. Now, purines and pyrimidines have the basic structure shown here. As you may remember from biology, adenine and guanine are purines, while cytosine, uracil, and thymine are pyrimidines. These 5 nitrogen heterocycles are the 5 nucleotide bases found in DNA and or RNA. I have a special personal way of distinguishing purines from pyrimidines. Structurally, I like to remember that purines are structurally larger. You'll notice that they have more atoms than pyrimidines in their core. The way I remember that is remembering that the word purine is smaller than the word pyrimidine. Thus, purine, which is a smaller word, corresponds to the structure that is larger, and pyrimidine, which is a larger word, corresponds to the structure that is smaller. Now, I realize that that might seem absolutely insane and like an extremely stupid way of remembering this, but for whatever reason, when I was learning these structures back as an undergraduate, that's the way I remembered, and it stuck. Now, in order to remember the names of the purines and pyrimidines, I remember that adenine and guanine, the 2 purines, begin with the letters A and G, which spell ag. Because I'm a USU Aggie, and Aggies are pure in heart, it's easy to remember that adenine and guanine, AG, ag, are purines, pure in heart. Now, the pyrimidines, cytosine, uracil, thiamine, begin with the letters CUT, or cut, while an AG, adenine and guanine, or ag, or Aggie, is pure. An infected cut, cytosine, uracil, or thymine, is not. Thus, our cut heterocycles are pyrimidines.